Okay. Hello guys, Dr. Rahul this side, and I will be discussing today biochemistry, right? So, okay, let me check once. It's recording. Okay. So first, I'll give the intro. How I will start with. First, I will discuss the inborn error of metabolism with few case discussions, correct? And then I will not discuss the cycles you will read, cycles you have to read by yourself, okay? But I will discuss the main points from this cycle, from where the questions are likely to be asked in your papers, correct? The things which you can read, I will not discuss that because I have to cover the session in quick time, because it's a revision session basically for you to look before exams, right? So from salient points I will cover, and also will cover the previous year questions that has been asked, right? And case scenarios will be there, right? So there will be many case scenarios, so be ready with it, right? Okay, so I will start, correct? Okay, so let us start. So first I will start with inborn errors of metabolism, right? It's a very important topic. This will collaborate with pedia a bit, right? But I will discuss it here only. Okay, so first scenario I will take about, say, in the question, if they give it like something like sepsis, okay, sepsis and hypoglycemia, sepsis plus hypoglycemia in a feeding baby. Mark my words, sepsis and hypoglycemia in a feeding baby. Means feeding baby means breastfeeding baby, right? So this case goes towards galactosemia, okay? Galactosemia. Now the important thing about classic galactosemia, which is usually asked in exam, that already been asked, okay? This I will be discussing classic galactosemia, okay? Classic galactosemia. Now here, the enzyme deficiency is asked is galput, okay? Galactose 1-phosphate uridyl transferase deficiency, correct? So in short, we tell this as galput, but often the full form is given galactose 1-phosphate uridyl transferase deficiency. So in this deficiency, there occurs decrease in glycogenolysis. Okay, so in this there occurs decrease in glycogenolysis, and also there is fasting hypoglycemia, as I have discussed. But main thing is that there occurs something known as oil droplet cataract. Okay, remember this one, oil droplet cataract. And treatment is non-lactose diet. Anyway, that is not important. These three is important, so these three don't forget. Classic galactosemia, galput deficiency, decreased glycogenolysis, and oil droplet cataract. Okay. Next, we will come to a condition known as hereditary fructose intolerance. Okay. Hereditary fructose intolerance. So this occurs more than 6 months. Okay. In weaning period more than 6 months, and there is no cataract. Okay. Treatment is fructose free diet. Treatment is fructose free diet more than 6 months and in weaning period. Okay, remember this one. This is about hereditary fructose intolerance. So by now, so far, what we have discussed. First, we have discussed classic galactosemia, galput deficiency, decreased glycogenolysis, fasting hypoglycemia, and oil droplet cataract. These are the features. Okay, and okay, and then we discussed about hereditary fructose intolerance, that is more than 6 months, weaning, and fructose free diet as a treatment. Correct. Okay. Now, next condition is secondary lactose intolerance. Secondary lactose intolerance, correct? So in this there will be a history of cow milk exposure. Okay, history of cow milk exposure, followed by persistent diarrhea, ठीक है, persistent diarrhea will be there, ठीक है, persistent diarrhea, and there will be perianal rash, ठीक है. This is mainly due to lactase enzyme deficient. Anyway, these three, this history, if there, then the case is going towards secondary lactose intolerance. Next, something about Hunter syndrome, ठीक है. So Hunter syndrome is basically, this is an MPS, right? Mucopolysaccharidosis disorder. This I will discuss separately, ठीक है. So here what will you find? Some features like macroglossia you will find, large tongue, okay, macroglossia. Then you can find gargoylic faces, okay. Then gargoylic faces may be there. See, remember hunter have clear cornea, so here cornea is clear, okay. And unlike other MPS which are autosomal recessive, hunters is X-linked recessive, okay. And males are affected. So remember, hunters are males and hunters have clear cornea, okay. This is the things which you'll find. And another thing is the enzyme that is deficient, alpha L sulfatase, right? And there are other MPS with no mental retardation, okay. What are those MPS? She's, which is MPS 1S, Morocco, which is MPS type 4, and Mortex Lemi, which is MPS type 6, correct? You may remember this. Alright, so now we will go to GSD, which is glycogen storage disease. Briefly I will describe later, but just to tell you about the salient points or the this clinical presentations, okay. So in GSD mainly what we will find, hypoglycemia, okay, hypoglycemia you will find. Then you will find lactic acidosis, very important, lactic acidosis you will find. Then you will find hepatosplenomegaly, renomegaly, okay, these features you will find. Then most common is von Jurki's disease, which is glucose 6 phosphatase deficiency, okay. So remember these points, okay. And okay, another important is Zellweger syndrome. So what we will find here? We will find here facial dysmorphism. We will find VSD, right? And then we will find hypotonia, floppiness, right? Is mainly due to accumulation of very long chain fatty acid accumulation, okay? Very long chain fatty acid accumulation due to alpha oxidation defect, okay. You can also tell it as paroxysmal protein target disorder. Anyway, just remember these points, this is enough. Due to alpha oxidation defect, there occurs very long chain fatty acid accumulation, okay. This is okay. This is actually paroxysmal protein target disorder, right? Next we will talk about important thing, Gaucher's. This has been repeated so many times. Gaucher's is very important. Now Gaucher's actually is a bone marrow disease, and that results in pancytopenia, thrombocytopenia, anemia, because it is affecting bone marrow, okay. There occurs elevated liver enzymes, there occurs hepatosplenomegaly, okay. There occurs organomegaly, liver enzymes are elevated, okay. Bleeding occurs, bone pain occurs, there occurs bone pain or crisis situation, and the enzyme defect is beta glucosidase defect. And the main important is important thing is there is a crumpled tissue paper appearance on bone marrow biopsy. So bone marrow biopsy shows crumpled tissue paper appearance. Crumpled tissue paper appearance. How it looks like? I'm showing you with a diagram. Okay. This looks like something like this. This like crumpled tissue paper. Okay. Something like this. Okay. Next. Now I will tell you some salient points which includes previous year questions and everything, right? So see, vitamin B12. Vitamin B12 is animal origin. Don't forget that, right? Usually we make mistakes in a hurry. Vitamin B12 always animal origin. This has been asked thrice. 3 times it has been asked. Vitamin B12 animal origin. Okay. Next, those who intake jowar, right? In them there occurs increase leucine that results in decrease NAD plus, which results in jowar intake causes decrease leucine that results in decrease NAD plus. Okay. That results in decreased niacin, and decreased niacin, what will it cause? Cazel's necklace. You know that Cazel's necklace. So Cazel's necklace, so many times it has been asked in picture based questions. Cazel's necklace. So I will show you how it looks like. It is actually in the neck region. Okay. Say this is the neck region. It will show something like I am showing you. Okay, this part will be black actually, right? This part will be black. Okay. Alright. So this is how it looks, Cazel's necklace, right? So I will show image in my separate sessions. Okay. Don't worry. Okay. So another thing that came, that glucagon stimulates hormone sensitive lipase, right? Don't forget this. Usually this actually the concept is dependent on insulin dependent hormones. Okay. So insulin dependent hormones, these are usually active after intake of food, right? In well fed state, glucagon usually stimulates hormone sensitive lipase. Insulin stimulates all hormones or all enzymes that are active in well fed state, is immediately after taking food. Okay. Now we will look towards sphingolipidosis, right? So often the enzyme deficiencies are asked, so we must be thorough with this. And all, remember that all enzyme deficiencies are autosomal recessive. Okay. Almost, except there are few exceptions, but mostly all enzyme deficiencies are autosomal recessive. Okay. So these are the diseases and these are the enzyme deficiencies. Please continue the table because I may not be able to write everything in this page. If you are taking the note, continue the table, right? So first is GM1 gangliosidosis, right? So here what is deficient? Beta galactosidase is deficient. GM1 gangliosidosis, beta galactosidase deficient. I'm continuing it, right? Tay-Sachs disease, beta hexosaminidase A defect, right? Tay-Sachs disease, beta hexosaminidase A defect. All right. Sandhoff disease, beta hexosaminidase A and B both defect. Okay. Then Krabbe's disease. See, these are asked in inicet, right? Match the following. Beta galactosidase defect. Beta galactosidase defect. ठीक है. चलो next. Niemann-Pick. So Niemann-Pick disease, sphingomyelinase defect. Is it sphingomyelinase defect? Just keep on noting it, guys, because there is no other options. You to remember it, right? Gotchers, I've already mentioned. This is very important. Don't ever forget this. This is the last NEET also. Alpha glucosidase defect, also known as leukocerebrosidase. Know both the names. Okay. Then Farber's ceramidase defect. Okay. Ceramidase defect, correct. Ullmann's acid lipase defect. Then comes Fabry's, which is alpha galactosidase defect. That's enough. So Zellweger syndrome, I will already mentioned that Zellweger syndrome is cerebro. See, again I am repeating it because 2-3 points are missed, so you should not miss that. This has been asked twice or thrice. Is important, right? Zellweger syndrome is cerebro-hepatorenal syndrome. Okay, cerebro-hepatorenal, 3 systems involved, right? Paroxysmal protein target disorder. Okay, and there is decreased peroxisomes, decrease very long chain fatty acid, and increase phytanic acid. A beta oxidation is blocked, therefore decrease very long chain fatty acid, increase phytanic acid due to inhibition of alpha oxidation. Okay. Then there is an important question that lipoprotein electrophoretic mobility. Take electrophoretic mobility. See, most mobile is HDL. Why? Because lipid content is less. Okay. Size is least. Density is maximum. Density maximum. Lipid less. Size is least. And ekdam slow is chylomicron because density is less but lipid is maximum. Okay. So it is heavy. It cannot move. HDL is light, so it will move. So after chylomicron there is LDL, VLDL, IDL. Simple. This is the electrophoretic mobility of all lipoproteins, right? Chalo next. Okay. Let's finish the enzymes, guys, because this is very important and I want to finish it first. Other things are other, we can finish it, we have much time, we can finish it later also. Okay, wait a second. Okay, so I will start with some conditions. Like, I'm in the enzyme section. Okay, classic homocystinuria, right? Classic homocystinuria. Why it occurs? Due to cystathionine beta synthase defect. Okay, and here cofactor is vitamin. Everything is important here, guys. You cannot miss anything. Classic homocystinuria due to cystathionine beta synthase defect. B6 is the cofactor. Everything has been asked. This has been asked. This has been asked. Okay, and there occurs subluxated lens. Okay, subluxated lens and ninhydrin is positive. Ninhydrin positive, but these two you may forget, but these two don't forget. B6 is the cofactor, cystathionine beta synthase defect. Okay, and there is something known as maple syrup urine disease. This is due to branch chain keto dehydrogenase defect. Okay, so what happens due to BCKD defect? The maple syrup urine disease, urine smells like maple syrup. Okay. Now next is oxalate stones. Why it occurs? Oxalate stones due to glyoxylate. Guys, don't miss these enzymes. It is very important. ग्लाइकोक्सिलेट एलानिन एमाइनो ट्रांसफरेज डिफेक्ट. ठीक है, चलो नेक्स्ट. आई एम टेकिंग द क्लास इन वेरी क्रिस्प फॉर्म. ठीक है, कार्सिनोइड ट्यूमर. ठीक है, कार्सिनोइड ट्यूमर, दिस again also very important. What happens there? First there is increase serotonin, only serotonin is increased. ठीक है, and increase 5 hydroxy indole acetic acid. ठीक है, and niacin is decreased. Okay, remember these 3 points. This is carcinoid tumor. So next, classic galactosemia I have described, Galput deficiency, oil droplet, cataract, right? And hereditary fructose intolerance in the weaning period, as I told, no, this is due to aldolase B defect. aldolase B defect in the weaning period. ठीक है, चलो. Now you may be, okay, let me check once. Okay. Now you may be given a case scenario like a patient came in your OPD with black colored sclera. ठीक है, black sclera, black urine. ठीक है, joint pain. ठीक है, or they may give something known as uchronosis, same only, uchronosis. ठीक है, what is the condition? They are talking about alkaptonuria. This is very common question, guys. Alkaptonuria, right? So here the enzyme deficient in alkaptonuria is homogentisate oxidase defect. ठीक है, and here silver nitrate test is positive. ठीक है, so alkaptonuria, patient will have black sclera, black urine, blackish things deposition in the joints, joint pain, uchronosis, AGN3 positive, homogentisate oxidase defect. Don't forget the enzyme homogentisate oxidase defect in alkaptonuria. ठीक है, urine on standing becomes black in color. ठीक है, चलो next. Then there will be something, hepatorenal syndrome. Just follow the lines. ठीक है, and then arrive the diagnosis. Hepatorenal syndrome. ठीक है, plus urine cabbage like order, urine cabbage. ठीक है, cabbage like order. ठीक है, and then succinyl acetone increase, fumarate acetoacetate hydrolase defect, right? Hydrolase defect. So what is the condition? The condition I am talking about, yes, correct, it is tyrosinemia. Fumarate acetate hydrolase defect, tyrosinemia, which one? Type 1. ठीक है, tyrosinemia type 1. Now talking about tyrosinemia type 2, which is mainly oculocutaneous manifestation, oculo and cutaneous, right? Tyrosine transaminase defect. Just remember tyrosinemia 2 मतलब TT 2T. ठीक है, tyrosine transaminase defect. ठीक है, चलो next. Now there is a very important condition, phenylketonuria. 5 star you can give. ठीक है, so here there is mousy order urine. Here every line is important. ठीक है, there is increase phenylalanine. ठीक है, increase phenyl acetate, increase phenyl lactate, increase phenyl pyruvate in blood. ठीक है, here FeCl3 is positive, good3 is positive. ठीक है, and this one enzyme deficient is dihydrobiopterin oxidase, sorry, dihydrobiopterin reductase, right? I will rectify this thing, dihydrobiopterin reductase defect. Don't forget this. Phenylketonuria, mousy order urine, increase phenylactate, increase phenyl pyruvate, increase phenylalanine in blood, dihydrobiopterin reductase defect. ठीक है, FeCl3 positive, good3 positive, right? Don't forget this point, right? Okay. Okay, so now we will talk about uh, some questions that has been asked. This one, type 2 GSD. This is also important, guys. So type 2 GSD is also known as Pompey's disease. Okay, Pompey's. Now actually it is a muscle GSD. Okay, this is muscle GSD. So this is your one hint. Another is the enzyme defect, which is lysosomal acid maltase defect. Okay, lysosomal acid maltase defect. Then I'm talking about GSDs now. Okay, you remember ABCD. ABCD mane for Anderson's disease branching enzyme defect. A for Anderson's, B for branching enzyme defect, C for Cory's, D for deep branching enzyme defect. So will you remember this, guys? Very easy. Anderson's disease for branching enzyme defect, Cory's disease for deep branching enzyme defect. Okay, so remember it up to this till now, and we will discuss it further, right? Okay, so there are some questions regarding the glycine products. This need also these are asked. Okay, this is also important. FMG is also these are asked. Okay, so first I will talk about glutathione. I will talk about the very important ones, and everything is not required. Okay, glutathione. Glutathione keeps your hemoglobin in reduced state. Okay, just remember this point that glutathione keeps your hemoglobin in reduced state. Another is arginine. Arginine, guys, is very important. 3 times it has been asked. So what are the compounds that is derived from arginine? This has been asked. One is nitric oxide, right? Next, urea. Next, ornithine. Next, agmantin. You can forget everything. Don't forget these two. This and this has been asked. Chalo. Yes. Another important question I saw, asked twice in UPSC. Measure and product. Okay. Measure and product of protein catabolism. Measure and product of protein catabolism. If I talk in humans, it is urea. If I talk about other mammals, then it answer will be allantoin. Okay, allantoin. Okay, now talking about tryptophan. This point is important, guys, so concentrate. That 60 milligram tryptophan will give 1 milligram niacin. Correct? This point is important because this is the only amino acid which gives a vitamin. Okay. Now, also you know that if tryptophan is deficient, then niacin will be deficient. Correct? And this will give rise to Caesar's necklace, as I have discussed earlier. And also, due to niacin deficiency, so many things happen. The 4Ds: diarrhea, dermatitis, dementia, death. Okay, these are 4Ds. These also happen. Anyway, yeah. So another thing is minkish disease. This is the one of the favorite topic of the examiner, particularly this one, UPSC. So actually it is ATP 7. I forgot A or B, so please do confirm this one. I guess it is A. Okay, if I am wrong, please do confirm once, because I am not able to remember whether it is A or B. So anyway, this confusion is between Minkies and Wilsons, right? One is A and one is B, correct? So you have to confirm this point. Okay. and another is this is lysyl oxidase defect. Okay, lysyl oxidase defect. This one please confirm, guys. Okay. Okay. Oh yeah, I have found the point. It is ATP7A only. Okay, ATP7A defect only, and copper is the this one cofactor here, right? It is the most. It is also important point, guys. So don't miss it, right? Now I'm talking about the structure of protein. This has also been asked in your FMG and UPSC CMS. Like in case of denaturation, there are many bonds: primary, secondary, tertiary, quaternary, many bonds. Primary is the peptide bond, and then there are hydrogen bond, covalent bond, etc., etc. Don't go in details. While we denature or heat the protein, secondary will be gone, tertiary gone, quaternary, everything will be gone, but primary will be retained. I will repeat again: when we denature a protein or when we heat a protein, primary structure is retained, everything is gone. Okay. Okay, now time to read about some collagen, guys. So collagen types and the sites where they are found, very important. If it is one, so remember one for bone. Okay, bone one. Okay, bone, ligaments, right? We'll find type 1. Okay, and type 2 where we'll find type 2 will find in cartilage. Okay, type 2 cartilage. Type 4 where we will find basement membrane, correct. If it is gone, it gives rise to Alport's syndrome, very important, guys. And then type 7 will find in skin. If it is gone, it gives rise to epidermolysis bullosa. Okay, or acquisita. Okay, whatever you say. Okay, so guys, I will be uh, summarizing whatever we have discussed till now. We will summarize. Okay, so just make some tables, right? Be ready with it. So here this is disease, and here I will write the enzyme is deficient. This is a 5-star table, and this is the all from this table you are going to get at least one question. So first disease is albinism. I will continue the table in the next page, right? Okay, so albinism, here the enzyme deficient is tyrosinase, correct. Then MSUD, I described already, BCKD defective. Now a branched chain keto acid dehydrogenase. Okay, and what are the components? Vil means valine, isoleucine, leucine. I repeat, valine, isoleucine, leucine. This is BCKD. Okay, so next I am continuing the table, right? Isovaleric aciduria. Okay, and its enzyme deficient is isovaleryl CoA dehydrogenase. Isovaleryl CoA dehydrogenase. Manage, guys, space is less, right? Please manage. Then homocystinuria, I have already discussed. Homocystinuria, cystathionine beta synthase, I already told B6 is the cofactor here. Okay, so next phenylketonuria, phenylalanine hydroxylase defective, also dihydrobiopterin reductase defective. This can be given as treatment as well. Okay, here what happens, the products I mentioned, phenylactate, due to deficiency of this one, this is the main enzyme. Okay, phenylalanine hydroxylase. Now this is due to deficiency of this enzyme, phenylactate, phenylpyruvate, these big compounds deposits in the blood and finally in the brain, and the brain lacks amino acids, so there occurs mental retardation as well. Are you getting my point? Due to the deficiency of phenylalanine hydroxylase in this condition, there occurs accumulation of phenylactate, phenylpyruvate, phenylacetate in blood, and they get accumulated in the brain, and there occurs lack of neutral amino acids in the brain that results in mental retardation. Correct? and their Guthrie test is positive, FeCl3 positive. Okay, don't forget this. This is important, guys. Okay, mental attraction is seen. Okay, chalo next point. Alkaptonuria I mentioned, homogeneity oxidase, tyrosinemia type 1, fumarate acetate hydrolase defect, tyrosine type 2, tyrosine transaminase defect. All that we have done. Okay. Now few tests and disease. These are also important, guys. Match the following can come. So don't miss, right? FeCl3, just now I mentioned, phenylketonuria, also for alkaptonuria. Just keep it in one place, right? Guthrie, you know, phenylketonuria, just now I mentioned. Okay. DNPH for maple syrup urine disease. Dinitrophenylalanine hydrolase. ठीक है, MSUD. चलो next. Overmeyer test. Overmeyer test means it is a PET test, in heart nerve disease. ठीक. Then cyanide nitroprusside test. Cyanide nitroprusside test. This is for homocystinuria. ठीक है, homocystinuria for diagnosis purpose, right? चलो. Vanillin mandelic acid. This is very important. FeO. And FeO rule of 10 is there. Okay, this I will discuss in surgery. Okay, not yet. So vanillin mandelic acid, FeO. Chalo next. 5-HIA. Just now I mentioned few slides ago. This is carcinoid. Okay, what happens here? Tell. Yeah, serotonin. What happens to serotonin? Increases. 5-HIA increases. What decreases? Niacin decreases. Good, right? Chalo next. Now few inborn error metabolisms with urine orders, guys. This particular slide is important for CMS because 3 times it has been asked from here. One is glutaric acidemia. Here sweaty feet order. Then Hawkinsinuria. ठीक है. Here swimming pool order. ठीक है. चलो next. MSUD, maple syrup order. I mentioned so many times. I think thrice as for now. No issues. Hypermethonemia. Repetition makes me, people perfect, right? So repeat as many times as you can. Hypermethonemia, boiled cabbage order. Guys, this has been asked twice in your CMS. Hypermethonemia, boiled cabbage order. Okay. And phenylketonuria, mouse order. Already told, right? Chalo. An amino acid that absorbs UV light. This has been asked. This is tryptophan. Okay, tryptophan. How much UV light? Around 490 nanometer. Okay, chalo. So next I will go to PCR. See, this slide is also important. Asked thrice in inicet. Like PCR, there are 3 processes. Okay, and you must not forget this. One is denaturation. Second step, annealing. Next, this one, extension. First, we have to denature. Okay, denature what? Double stranded DNA. Okay, to denature it more than 90 degrees Celsius. Each and every word is important here. Annealing with a primer 60 degrees Celsius and extension 72 degrees Celsius. Am I clear? Here enzyme is used. Enzyme has been asked twice. Taq polymerase. This has been asked in AIMS June also. 2020, right? Taq polymerase is the enzyme for extension. Okay. Now there's another important concept about DNA repair defect. This has been asked in recent NEET, right? this neet that occurred few months ago, no, this neet 11 September wala. So one is nucleotide excision repair. I will write short form, but you please write the long ones, right? Otherwise you'll be confused. So I will repeat twice or thrice so that you don't have any problem. This is a boring subject, guys, but you have to cope up with this. Nothing can be done, we have to study. Nucleotide excision repair, right? So for example, xeroderma pigmentosa, trichothiodystrophy, right? Cocaine dystrophy. Cocaine spelling is like this, cocaine. Okay, cocaine dystrophy. I will repeat. Nucleotide excision repair, xeroderma pigmentosa, it is very important. This is only asked, right? Cocaine dystrophy, trichothiodystrophy, don't forget this one, right? So next, base excision repair. I will again say base excision repair, mutation of polyposis, mutated polyposis. Okay. Then mismatch repair. This one came in last neet. Mismatch repair. This HNPCC. Okay. Double stranded DNA break repair. Skid. What is skid? सबएक्यूट combined immunodeficiency. ठीक है, चलो. Let's see some RNA polymerase eukaryotic. This is not been asked, but maybe asked in future. I feel, I have a gut feeling like this will may be asked. Just remember, this is easy only. Eukaryotic RNA polymerase. ठीक है. Type 1, type 2, type 3. Type 1 gives ribosomal RNA. Type 2 gives all types, mRNA, miRNA, snRNA, ठीक है, सर्कुलर RNA. And type 3 gives siRNA. ठीक है, type 3 gives tRNA. Remember, this thing is enough. Okay, another thing, guys, they ask about tests for DNA, RNA, protein. So I will make it clear here only. I will give you a mnemonic, you will not forget ever, right? DNA, RNA, protein. So how will you detect these things, correct? So actually this is a blotting test. With what blotting test you can detect DNA, RNA, protein? So guys, I will use another color. See, I made a short form like DNA. This is dosa, this is roti, and this is pizza. Okay, so DNA is dosa. Dosa is taken in south mainly. So with south blotting test you can get DNA. Roti is taken mainly in north, so in north blotting you can detect RNA. Protein, pizza is mainly taken in the west, so in west blotting you can detect protein. Okay, so this is only please don't say this to anyone, like it is a joke type thing, but and don't feel offended because of the food items. This is just for mnemonic, right? Just for the purpose of mnemonic, just for you to remember, right? So yeah, so dosa, I mean DNA for southern blotting, RNA for northern blotting, protein western blotting. Now we will not forget, right? Let me remove it. I mean, my mnemonics are too bad. Okay. Okay, chalo, next. Next what? Yes, now we will study some DNA enzyme and their function. Okay. See, helicase. What is the function of helicase? This has been asked. Okay. It unwinds DNA. First function. Second function, it breaks hydrogen bond. Unwinds DNA, breaks hydrogen bond. Don't forget this. This has been asked. Next, what is asked is ligase. Should I change the color? Yeah, it's better. So ligase. Ligase for sealing nicks. Okay? This has been asked. Next, topoisomerase. Topoisomerase is a nicking and resealing enzyme. Okay? So next. Now we'll talk about glucose transporters, right? This is important, guys. Okay? So see, 1 and 3 both are present in brain, but 3 is in the neurons of the brain. A simple word here and there changes everything. The answer will be completely changing. So be careful. 1 and 3 present in brain, but 3 is present in the neurons. Okay? 2 is present in the intestine. Okay? Now 4 is very important. Give me some time. 5 in testis, fructose testis. Okay? 6 in spleen, 7 in liver. Right? Never asked. This one is asked. Take another pen. Okay. This is asked because this is insulin dependent. That's why it is asked. Just remember HAM. So GLUT4 is HAM. What is HAM? H for heart, adipose tissue, and skeletal muscle. Insulin dependent GLUT transporter. You remember this? This one is insulin dependent. HAM, heart, adipose tissue, skeletal muscle. This has been asked thrice. Chalo. Now inhibition. This is very, very important. More than 5 times repeated. Also asked in 2020 NEET. Okay. So competitive inhibition, uncompetitive inhibition. Guys, follow the word non-competitive inhibition. Right? So just learn in a short way. Calm down because this is confusing. Vmax and Km I'm writing here. Okay. Correct? Done? Now competitive in C. Vmax is not the issue, right? Vmax you can rule out, right? Problem is in the Km. So just remember Km is increased in competitive, Km is decreased in uncompetitive, no change in non-competitive. You remember one, rest you can rule out. Just remember this one. Okay? Km increased in competitive, Km decreased in uncompetitive, Km no change in non-competitive. That's it. Because confusion occurs in this one. If you remember, try to remember both, you will end up missing the question. Okay, and now we will go to DNA. I will give you just key points. Watson-Crick base pair rule that tells us that A double bond T, G triple bond C. This is hydrogen bond. This is hydrogen bond. But with adenosine and thymine double bond, guanine and cytosine is triple bond. Remember this. Correct. Next important point is salvage pathway. Repeated twice. Okay, salvage pathway. See, here the important thing is Lesch-Nyhan syndrome. Don't forget this HGPRTase defect. In this case there is self-inflicted suicidal wounds. ठीक है, चलो GSD you have done, right? GSD1 I have mentioned that it is glucose 6-phosphorase defect, right? Von Jarkis. Now I am classifying this, right? Von Jarkis glucose 6-phosphatase defect. Okay, and number 2 is, okay, let me next page. Number 2 core is, sorry, it's number, not number 2, number 3. There is no number 2, guys. There is no number 2 GSD. Number 1 GSD Von Jarkis glucose 6-phosphatase defect, and number 3 GSD it is Coris. Number 4 Anderson's. Number 6 RAHUL'S. Number 7 Darwis. Don't worry, I will describe each of this. Don't worry. Okay. So Coris I already described this way, right? Branching enzyme defect Anderson's, Coris deep branching enzyme defect, these I have discussed. Number 6 is RAHUL'S. RAHUL'S mane liver phosphorylase defect. Liver phosphorylase defect, not so important. Now see, muscle GSD plus exercise intolerance. Now there will be with hypertrophic cardiomyopathy. I have told you, muscle involvement, it is type 2. Type 2 is Pompis. That's why I did not mention that earlier. Earlier I didn't want to mention type 2 because I want to mention it here. Type 2 is Pompis, correct? With this one hypertrophic cardiomyopathy, only muscle involvement, right? And without hypertrophic cardiomyopathy, only exercise intolerance, type 5 and type 7. Type 5 is McArdle's, type 7 is Darwis. Okay. Now type 5 muscle phosphorylase defect and type 7 erythrocyte phosphofructokinase defect. This is not that much important. This one is important. Type 2 pump is hypertrophic cardiomyopathy. This one don't forget, right? It is in this one muscle, you just remember muscle GSD, pump is hypertrophic cardiomyopathy type 2. That's it. This is very important, right? Don't forget this. This one is all right. I've never seen that coming. Okay. So always read type 2 separately, right? Don't mix in that, because GSD only type 1 and type 2 is important, and rest I've never seen coming, right? So type 2 always read separately. Don't forget. Chalo. Okay. So now what will we do? We will learn some vitamins, correct, and their deficiencies. Like first I will take pyridoxine, which is B6. This has been repeated twice or thrice. Last time I said only came. This deficiency will cause seizures. Okay. Peripheral neuropathy. Okay. Homocystinuria. Because last in last few slides ago I told homocystinuria due to cystathionine beta synthase defect. B6 is the cofactor. Told you, no? Folic acid. Yes, deficiency will cause. This came in this neat. Megaloblastic anemia, correct? subacute combined degeneration, right? Homocystinuria, yes, may happen. Now some vitamins and their coenzymes, yes, this is also important. Thiamine, transketolase, ठीक है. Niacin dehydrogenase, right? Isolated dehydrogenase. Then cobalamin, methyl malonyl CoA mutase, ठीक है. These are the coenzymes, right? And most important, biotin. Always remember box reaction, box biotin carboxylation reaction, and here carbon dioxide and ATP is required. This has been asked so many times. Biotin carboxylation reaction, carbon dioxide, ATP required, box reaction as I say. So here coenzyme is propionyl CoA carboxylase, because carboxylation reaction, so carboxylase. Okay, don't forget. Okay, box reaction, box, right? Now urea cycle disease, so important. Cycle, you remember, I will tell you the disease and the enzymes, right? So CPS1, if it is deficient, CPS1 is carbamyl phosphate synthase 1, okay? If it is deficient, gives rise to hyperammonemia type 1. Please remember the substrate NAG, N-acetyl glutamate, very important. Has been asked, this has been asked, this has been asked. Okay, chalo. And OTC, deficient hyperammonemia type 2, ठीक है. चलो, arginocyacinate synthetase deficient citrullinemia, ठीक है. arginocyacinate lyase deficient arginosuccinic aciduria. Something peculiar here happen. This has been asked because it is peculiar, of course, that's why. A hair change, trichorhexis nodosa, don't forget this asked. Arginosuccinate lyase defect, arginosuccinic aciduria, trichorhexis nodosa, ठीक है. चलो. And arginase, arginemia, never asked. Anyway, now again few vitamin topics left. Vitamin E deficiency will give rise to hemolytic anemia, this is important, guys, and peripheral neuropathy. Thiamine deficiency, very, ठीक है. Niacin deficiency, you know, pellagra, casus necklace. B5 or pantothenic deficiency, yes, this is important, this has been asked recently. Pantothenic acid or B5 deficiency, nutritional melalgia, burning foot, also known as burning foot, okay, this is important, has been asked. Chalo. Now one thing is that keep attention, right? This one, vitamin E deficiency causes hemolytic anemia, correct? But vitamin K toxicity causes hemolytic anemia. Vitamin E deficiency causes hemolytic anemia, vitamin K toxicity causes hemolytic anemia, correct? These are the tricky points for the examiner's trick, you know. We will not let them trick this time. Joking, right? They will trick every time. Anyway, is something left? Yes. Enzyme and allosteric activators. Also a repeat topic, as you know. Acetyl CoA carboxylase. For this enzyme, citrate is the allosteric activator. This has came in 2020 AIMS. Okay. For CPS1 NAG, just I mentioned few slides ago. For pyruvate carboxylase acetyl CoA, correct. For PFK1, it is fructose 2,6 bisphosphate, correct. CPS1 NAG, pyruvate carboxylase acetyl CoA, PFK1, fructose 2,6 bisphosphate, correct. So we have completed major topics. Now I will take the jaundice section here only. Okay. Two types, prehepatic, posthepatic. ठीक. Pre means unconjugated and post mane conjugated, because prehepatic means prehepatic before the liver, posthepatic means after the liver, means there is a block. So block mane conjugation. Block means after the bilirubin is conjugated, right? So obviously this conjugated bilirubin will increase. Okay, let me rub it. So just simply remember, in unconjugated thing, or this one prehepatic, total bilirubin will increase, and unconjugated bilirubin or indirect bilirubin will increase. Direct bilirubin will be normal, but here direct bilirubin will increase. Okay, here urobilinogen will be there, that's it. But here conjugated bilirubin will be there in urine and this one bile and everywhere, right? Conjugated bilirubin, direct bilirubin will increase. Hayes test, Foches test, everything will be positive in this case. This only, not much. And lastly I will discuss TCA cycle. I will not discuss, I will write the sequence of the 8 enzymes. Never asked, but maybe asked sequence, because in inicet sequence are asked. So first is citrate synthase, aconitase, isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, succinate thiokinase. I am following, guys. Succinate dehydrogenase, fumarase, malic dehydrogenase. ठीक है, 8 enzymes in sequence. ठीक है, don't believe anything. Some things will be left, but majority things are covered. Okay, let me check once. Okay, remember one thing, that in Niemann-Pick disease we will fight cherry red spot, also foamy macrophage. With this I think I've covered major part, considering the revision segment. If you find any difficulties anywhere, just drop in the comment box, let me know, and I will take it briefly. Okay, best of luck. Thank you. Join me in the channel and put your comments. Let me know what you want me to discuss, in my next sessions, right? Till then, okay, good night.